Thank you very much for attending our presentation. I would like to introduce to you today Apologix. Apologix is a biotech company in the oncology field. We develop monoclonal antibodies, and we're a spin-off from the University of Geneva. The company has so far received incubation support from Fondation Eclosion, the Geneva Life Sciences Incubator, VentureKick, and InnoSwiss. And I would like to point out at this point that we are the winners of Stage 3 VentureKick startup competition. This is the most prestigious startup venture competition in Switzerland. We start our journey with two projects in license from the University of Geneva, two monoclonal antibodies, code name H225 and 9F8, indicated for different cancer indications. I will spend the rest of my presentation on the first asset, monoclonal antibody H225, indicated for B-cell lymphomas, because it's the asset we're really spending most of our time working on. This product, it blocks the function of a protein called junction addition molecule C, JAMC. What is a JAMC? JAMC is a protein that localizes to attack junctions of endothelial cells. It's expressed by certain B lymphocytes, and also certain lymphoma cells also express JAMC. And there is a rationale whereby by blocking JAMC, you can then block the migration of lymphoma cells through the bloodstream into the target organs, which are spleen, liver, and others. Now, JAMC is not expressed in all lymphomas. Here we took 250 patients, we took malignant B cells from them, and we tested whether they were JAMC positive or negative. And we can see that in the case of mantle cell lymphoma, HCL, and some other indications, a large percentage of the patients are JAMC positive, but in other lymphomas, they are not. Therefore, we have very targeted therapy where we can go after those patients that express JAMC and not those patients that do not express JAMC. Based on these results and our preliminary experiments, we've decided to focus the development of H225 in the following indications: mantle cell lymphoma, marginal zone B-cell lymphoma, and CLL. Why? Because these are indications where there is a large unmet medical need for the treatment of patients who relapse after a first or even second-line treatment. For example, rituximab is first-line treatment most of the times for these patients, BTK inhibitors also, but the reality is that most of these patients eventually do not respond to these treatments, and because of that, there is the need for additional therapies. In this experiment, there is a classical overall survival experiment. What we do here is we take mice, we inject them at time 0 with JAK1 cells, which are mantle cell lymphoma cells, and then at day 5, we treat them with either a control antibody, H225, or the gold standard in the treatment of mantle cell lymphoma patients, rituximab. And we can see, of course, that when we're treating with a control antibody, after 40 days, all of the animals are dead. But when we treat with either rituximab or our own antibody, they live for much longer. In this case, rituximab was superior to our antibody, and this is not surprising because we have not really optimized the concentration or the dose regimen of our antibody. We can also see here what happens to spleens. We can see, for example, if we take spleen biopsies with animals treated with a control antibody, we have large spleens. In animals treated with our antibody, we have much smaller spleens. What does that mean? That means that the lymphoma cells have failed to travel to the target organ, in this case spleen, and because of that, the spleens are simply much smaller. This is also shown here where we took a look at for example, let's take a look again at spleen. We could take a look at liver, bone marrow, or lymph nodes, which are the target organs for lymphoma, but let's take a look at spleen. For example, animals treated with a control antibody, the percentage of tumor cells goes from 40 to 80%, while animals treated with our antibody, the percentage of tumor cells is almost down to 0. And what this clearly shows to us is that by blocking JAMC, we are preventing these lymphoma cells from traveling through the bloodstream into the target organ, in this case, spleen. Our development plan is a classical development plan. We're trying now to raise 2.5 million euros, Swiss francs, for the initial proof of concept studies and the initial toxicology studies that will take us to this value inflection point where we'll have a non-clinical package finalized. And beyond that, we plan to raise first 6 and then 12 million euros to take our product to market. We plan to use accelerated pathway approval mechanisms and other ways to really, with very few clinical trials, be capable of getting product approval within a reasonable amount of time. The team is made up of myself, the founders, Ignacio Faust. I have a PhD in biochemistry and management experience. Professor Imhoff and Professor Mathes are also co-founders, professors at the University of Geneva, and we're building a world-class management team that has expertise in the preclinical and clinical development of antibodies. Thank you very much for your attention.
My name is Diana Moore and I'm the CEO and co-founder of Move On Therapeutics, a biotech spin-off from the University of Zurich with the mission to advance a personalized cell therapy for skeletal muscle regeneration. There is a constant decrease of the regenerative potential of skeletal muscle tissue with age. A prominent example of a highly debilitating disease caused mainly by a damaged small skeletal muscle tissue is stress urinary incontinence. It represents the involuntary loss of urine by increased abdominal pressure. The disease affects over 200 million people worldwide, and the dark numbers of this taboo disease are much higher. 75% of the affected patients are women. Every fourth woman is affected after childbirth. And importantly, it's not only elderly people's disease, because 40% of women over 40 suffer from stress urinary incontinence. The market is huge, and the comorbidities associated to this disease are numerous, ending up even to severe depression cases. Now let's have a look at some real-life situations. Significant number of the women in your life might feel like this. It might be your mother, your wife, your daughter, your friend. Guess what they have? They have received the current gold standard therapy for moderate to severe urinary incontinence and may have experienced some of the potential severe side effects associated with the invasive surgical procedure. And this is the wall we will break. No one shall suffer in silence because of lack of a safe and effective long-term treatment for the underlying problem, namely a damaged muscle. We at Move On have the mission of changing the life of our patients for real, helping people regain control of life by means of regenerative medicine. See how one of our patients is enjoying life to the fullest again. Importantly, this impacts also the lives of her family, friends, and co-workers, having her again as an active part of their lives. We obtain a lower leg muscle biopsy, isolating growth on muscle precursor cells under strictly regulated GMP and xeno-free conditions, then inject the cells into the weakened sphincter muscle of the patient. Adjacent stimulation enhances the regeneration and functional restoration. Now, how do we plan to scale up to be able to capture the skeletal muscle regeneration market to bring the various therapies from our platform technology efficiently to patients? The core component of our business model is the development of a robust, automated production process validated for use in a C-class clean room. Through strategic partnering, production hubs can be set up around the world, each of them producing therapies for the surrounding healthcare ecosystems. Potential partners for our first asset are already identified, and they include larger pharma and biotech companies that are active in urogynecology and also show an interest in cell therapies, as well as the growing number of CMOs that are establishing themselves in the cell therapy space. Thus, enabling Move On to act as an early-stage accelerator for bringing innovative therapies to the clinics. My team of experienced researchers and physicians managed the successful translation of process from the bench to the clinics, supported by 6.5 million by the European Commission. Last year, we successfully completed our phase 1 clinical trial and secured another 6 million in funding for our phase 2. Now, we are actively fundraising 18 million, targeting the incorporation of high-end automation in the production process for value chain optimization and the clinical development of further pipeline indications. This is just the beginning of unlocking the potential of regenerative medicine. To date, personalized cell therapies have mainly been developed for deadly diseases. Move On wants to change this paradigm and bring personalized cell therapy to patients with conditions that, while non-life-threatening, are highly debilitating and significantly affect day-to-day living.
Hello, everybody. I'm Tino Matter from Anaval Medical. We tackle seroma formation, an issue that affects millions of patients worldwide every year. One of these patients is Victoria. Victoria was diagnosed with breast cancer at age 36. It was a routine procedure where surgeons removed part of her breast to fight the cancer, and she was able to recover at home. 1 week after the surgery, though, she felt pressure building up under her sutures and liquid started to explode through her stitches. Alarmed, she went to the hospital, where they diagnosed her with seroma, a chronic accumulation of liquid under the skin. She had to go back to the hospital over and over again to get her seroma treated, after all the emotional and physical stress she had to handle. Or, as she puts it herself, it was really bad. The last thing you want to do is to go back to the hospital for yet another needle. Victoria is not alone. There's millions of patients just like her every year. This is what a seroma looks like. It's this bulb of liquid that forms right after surgery where tissue has been removed. What is done nowadays sounds archaic because the surgeon just sticks a thick needle into the seroma, sucks out the liquid and hopes that it doesn't come back. Well, more often than not, it does come back, and you have to aspirate over and over again, on average 3 to 7 times. Along this way, you're faced with a delayed cancer therapy, with a fibrosis risk and an infection risk. In an overarching collaboration between scientists and leading surgeons in the field, we have developed a solution based on cutting-edge nanotechnology to solve this issue. Our nanoparticles are injected into the seroma one single time to make the seroma heal. In this way, helping the patient to recover faster, doing so cheaper than the standard of care, and in this way, getting happy patients and happy surgeons. We have shown in our animal models that the particles don't have any tissue toxicity, they don't clear to organs, and they don't have any systemic effects, meaning that they just unfold their activity where they are put in the seromas. And the seromas, they heal with a 100% rate, as opposed to the standard of care, which is around 35%, and fibrin glue, which is around 50%, which is something that surgeons sometimes use to make seromas disappear. So they have very superior seroma healing effects. Additionally, after 6 weeks, we looked at the surgical site, and opposed to standard of care and fibrin glue, you can see that the surgical site that has been treated with our nanoparticles, there you can barely see that an incision has been made because the wound healing was accelerated that much that you can barely see a wound there anymore. We are looking at the breast cancer field on the one hand side, which are mastectomies and axillary lymph node dissections, where we are looking at the 500 million target opportunity for the treatment of seromas that occur there. On the other hand side, on the prevention side, we have abdominoplasties that are another half billion US dollar market opportunity. And we plan to sell our product as a single-use device to hospitals. Who is we? I'm Tino Matter. I'm the co-developer of this technology, and I've always been passionate about innovating materials to improve people's lives. I'm very honored and happy to see this invention going to the market. With me is Sebastian Loy, who has a business background and a very strong entrepreneurial drive. We are completed by a senior team of researchers and a competent clinical, scientific, and business advisory board. Our regulatory pathway is quite clear. This quarter, we're going to verify our Class 3 classification with the FDA, start large animal trials in sheep, and then by 2023, we plan to start our clinical trials and then obtain market approval by 2026 in the US. Due to the fact that there's a clear medical device pathway, there's a better reimbursement, and it's a larger market. Additionally, it's quite a concentrated market with 20% of hospitals performing 65% of procedures. And there we plan to start after approval with a couple of showcases with hospitals that we already talked to now. And then by 2027 have our first sales in the US. Then we're going to expand by 2028 to other markets. We have reached quite a lot with non-dilutive funding so far, with 1.5 million from different sources, and we are now raising a seed round in June this year. You're very welcome to join that. And just one last thing, we have these properties of our nanoparticles that I've shown you. They're very regenerative, they heal wounds quite fast, and these features are applicable to a wide range of different wound healing scenarios. So we really see this technology as a platform technology. With this, I would like to close. Thank you very much. Contact me if you're interested. Thank you.
Hello and welcome to our presentation. My name is Damien Lotery and I am the co-founder and CEO of Readily 3D. At Readily 3D, we develop a new 3D printing process that is orders of magnitude faster and more convenient than traditional layer-by-layer 3D printing. It's called volumetric printing. It prints the entire object at once in 30 seconds inside a sealed, sterile glass container directly in suspension in a photoresin. We have a product that works and which we sell. We achieved break-even results last year with a forecasted net positive result this year. Our current focus is the market of biofabrication. Biofabrication is 3D printing for life scientists. It is used to make artificial models for studies and drug screening. It's used heavily in academic labs with a fast-growing user base in pharmaceutical and clinical settings. Our printers open up new use cases that were previously out of reach for researchers. The process is gentle, so it can print fragile cells. It's precise, so you can print complex structures, and it's fast, so that you have the ability to scale up your experiments. Besides biofabrication, we are also looking at applications in audiology and dentistry. Here, we empower practitioners to make customized parts, like hearing aids or clear aligners, directly in front of their patients, instead of dealing with days or weeks or turnaround times. Together, these markets provide an opportunity worth hundreds of millions of dollars. We started with the bioprinting market because we had a strong pool and a zero time to market. Now we want to grow our sales there and expand to audiology and dentistry, which are bigger markets. They are also more challenging due to the material certifications, high volumes, and the distribution channels, which are tightly controlled by a few players. Our USPs here are the speed and simplicity of the process, which enable in-shop printing. We have several competitors in the markets we're addressing, but almost all of them are using layer-by-layer technology. In comparison, we have a patent-pending solution that's faster, yet precise, and with a new choice of materials and geometries. Today, our product range includes the TomoLite volumetric printer, the corresponding software apparat, and a set of consumables such as vials. Currently, we make most of our revenue from direct sale of printers. Our plan in the future is to reduce the price point of the printer and shift more of the revenue towards the recurring sale of consumables and ink. This technology is patented and is exclusively licensed to us. Our software is proprietary as well. Our team currently has 8 employees, with an additional salesperson and computer scientist expected to join soon. The founders are myself, Paul, and Chris from the EPFL microengineering department, and we are advised by our seed investors who are themselves entrepreneurs in similar businesses. This project started in 2017. Just 3 years ago, we launched our first prototype, and last year we've made our first sales. We should be making our first cumulative million of revenue in the coming months. Our immediate focus is to reduce the price point of the printer and to increase sales volumes. We're also making a modular platform that will adapt to special customer demands and which will be a stepping stone for the next step, which is to break ground in audiology and dental 3D printing. What we are looking for is to raise $3 million to accelerate sales in bioprinting and to fund our development for the audiology market. Please get in touch with us if you would like to know more, and let's push the frontiers of biofabrication together. Thank you for your attention.
Hello everyone, this is Luisa Dobre, founder and CEO of Comet Health, a young company based in Zurich, Switzerland. At Comet Health, we are dedicated to transform the way healthcare professionals communicate and collaborate, helping them take fast and informed decisions, ultimately saving lives. As a matter of fact, an efficient communication is a contributing factor to 250,000 deaths every year. And now let's take a step back and understand why this happened. If you would be a doctor and you would treat a patient who is hospitalized for 4 days, you might need to interact with 50 other healthcare professionals from different specialization shifts, nursing staff, administrators, and so on. And the only way to communicate with your colleagues is via very ancient ways of communication, like pagers, first generation mobile phones, fax, switchboards, paper. And since you have a mobile phone in your pocket, you would use it to use commercial messaging applications like WhatsApp, which are unsecure. All this low efficiency, lack of integration, lack of compliance leads to patient errors. So that's why we have developed a communication and collaboration platform similar to Slack, but purposely built for healthcare. Our application is meant to integrate seamlessly into existing healthcare systems like electronic medical record, lab system, radiology systems, and so on. Not to duplicate what is in those systems, but to trigger notifications to push actionable real-time patient data at the fingertips of healthcare professionals. In a nutshell, push the right information to the right user at the right time on the device of choice, being at a mobile or a desktop computer. Our platform is built to become the all-in-one secure communication hub, one single interface, aggregating data from a variety of different sources in the form of notifications. For healthcare, it's crucial to put the patient at the center of conversation and giving providers the context, automation, and information they need for them to take the right decision. That's why we have developed a patient-centric messaging platform, which can be integrated with the EMR and is setting up a new standards in healthcare and dramatically improving the patient care. There are a zillion of secure messaging platforms out there, but the only way a messaging platform can provide utility in healthcare, it's crucial for it to offer context, clarity, and convenience, and this is what we are doing at Comet Health. We are basically giving back the time to the nurses, allowing them to spend more time with the patient and less time with administrative tasks. We help them save 3 to 4 hours a day, reducing interruptions by 80%. So far, our platform has been implemented in 10 hospitals, and by end of this year, we will reach an annual recurring revenue value with these contracts of a bit more than $500,000. So far, we have 4,000 active users on our platform and a solid sales pipeline of close to 400 sales opportunities. In order for us to reduce the sales cycle and accelerate growth, we are integrating with electronic medical records, middleware providers, telecom, and so on. At the moment, we are integrating with the biggest electronic medical record provider here in Switzerland, which through one integration, we can afterwards replicate it in all their hospitals where they are implemented and reduce the sales cycle up to 80%. Our platform will be used by more than 500,000 healthcare professionals in 6 years from now, generating revenues greater than 40 million per year with multiple exit options. We are now raising a pre-series A of €1.5 million in order to integrate with key partners, as we have mentioned before, expand to Germany, Austria, and Italy, and further develop the product. So far, we have raised $2.5 million from private investors and family offices. We have a dedicated winning team of seasoned entrepreneurs, business leaders, brilliant engineers, and healthcare professionals, and we are all committed to revolutionize communication in healthcare. Please reach out if you would be interested to learn more. Thank you very much for your attention, and I wish you a great conference.
I have founded Exploris Health in order to enable the long-sought transformation in healthcare. Heart attacks are the #1 cause of death. The current evaluation of coronary artery disease is not efficient. Too many patients undergo unnecessary examinations, and patients at high risk are not detected.
The Cardio Explorer test detects potentially life-threatening constriction of heart arteries, which is the main cause of heart attacks. We are among the first companies in the world who have validated a CE-marked medical AI test in 3 clinical studies on more than 2,000 patients. Our test uses standard data, which is already available for initial examinations. However, the accuracy is on the same level as the far more complex and expensive imaging procedures only available in specialized clinics.
We are pioneers in artificial intelligence, and we have developed a proprietary, highly automated modeling, optimization, and decision-making engine which exceeds the capability of commercially available software. Therefore, we combine several artificial intelligence methods in a holistic approach, allowing to reveal more relevant information in patient data.
Cardio Explorer is the perfect tool for the initial assessment in patients with suspected coronary artery disease, as it provides equal accuracy as current standard imaging.
We have all the expertise on board. We have a clinical team of leading cardiologists who are experts in AI-based medicine. We have a team of leading AI modelers, a pro in account management. We have deep experience in business development, marketing and communications, and last but not least, our head, Peter, the digital health entrepreneur. We all love the startup environment where we have the chance to shape a product, shape a company, and even a whole industry.
Join us, bring cardio diagnostics to a new level and save lives.